St. Mark's School of Texas is a private, all-boys school in Dallas, Texas. It holds at its core values that make boys into men. These values include leadership, integrity, courage, and honor. Its mission is to foster a community that creates young leaders who are able to go out into the world and make it a better place. To do so, the learning system has evolved into one of discussion, investigation, and exploration. The leaders that St. Mark's makes are able to go out in the world using what they know from St. Mark's in order to be more effective leaders both at home, in the workplace, and around their communities.
The SM Texas iGEM team was established to provide an outlet for boys at St. Mark's School of Texas to conduct independent research in the biotechnology fields while at school.
This past year, a senior, Halbert By, comes up to me and said, "Hey, let's get iGEM going." And I said, "Oh, okay, let's do it." And he did it all. He established it. He integrated with the biology club and said, "Hey, biology club members, this is a competition that we can do." And what I think what sets us apart is the cohesiveness of this group to be able to work so well together. This group is really, really good at problem solving and finding the answers to a lot of questions. The way that Halbert has set it up is we have four different committees, and they work together very well, and there's a lot of some of the students jumping from committee to committee, which is good, but their ability to collaborate and bounce ideas off each other and to be, once some students show up, the other students by saying, "Oh, well, I see your point, but well, we got to think of this." And so there's that constant dialogue going on, and that dialogue is what drives the team's progress.
Hello, my name is Killian Green, and I'm Director of Outreach for the St. Mark's iGEM team. This year's iGEM team has really improved and augmented my interest in science and biotechnology. I'd always really liked science and biology, and with this year's freshman biology course, I decided, why not try biology club and our iGEM team? So I went the first couple of times, and I found a lot of faces that I liked and I hung out with, and not only found those people that I connected with, but I also found the material and the things that we were doing really quite fascinating.
Lung cancer. It is a cancer that kills the most people in the United States. It kills more people than breast cancer, colon cancer, and pancreatic cancer combined. In 2014, almost 160,000 Americans are expected to die from lung cancer, accounting for almost 27% of all cancer-related deaths. Lung cancer, a very potent, malicious disease. But recently, Halbert, after seeing the experience of a service dog, has found a way to utilize the sense of smell of the dog to fight this disease.
While a variety of mechanical and electronic noses have been developing the past few decades, we propose genetically modified bacteria, or E. coli, used in conjunction with a mechanical device to detect lung cancer sensitively, inexpensively, and non-invasively.
The ALDB gene codes for a functional aldehyde dehydrogenase, which is directly induced by ethanol. Metabolism of the alcohol consists of its transformation into an aldehyde and then into a carboxylic acid, and activating several related pathways. The acid reacts with a BAR-A histidine sensory kinase, a signaling enzyme involved in a two-component signal transduction system present in E. coli. To catalyze the breakdown of various carboxylic acids, the kinase is additionally responsible for the induction of ARPOS, a regulatory gene of ALDB that directly opposes FIS during the activation of the ALDB operon. Eventually, BAR-A triggers a series of vital reactions that affect the CRP-CAMP regulatory mechanism, a dual complex that controls the expression of the ALDB coding sequence. In the complex, CAMP conforms the shape of CRP, also known as CAP, catabolite activator protein. This newly conformed CRP then attaches to the promoter and contributes to the initiation of transcription of the ALDB operon. Downstream of ALDB, CFP, cyan fluorescent protein, is expressed and the bacteria exhibits cyan fluorescence.
We use the FRMR gene to indicate the presence of the VOC formaldehyde. The way this works in the bacteria cell was that FRMR, the regulatory gene, would transcribe a regulatory protein. Also along the line, the protein would bind to an operator and thus make RNA polymerase unable to move down the line to express the gene GFP, which would make our bacteria glow. Even though there would be a promoter down the line as well that would increase the affinity of RNA polymerase in the DNA strand, the presence of this repressor, which is the protein, will not allow the GFP to be expressed. But if we introduce formaldehyde into the system, what would happen is that the formaldehyde would bind with the protein and act as a co-repressor. And in this way, the protein will become inactive, and then the RNA polymerase will be able to move down the line, the GFP, making bacteria glow green. Thus, if formaldehyde is present in the cell, then the bacteria will glow green.
The genetically related expression of the XYLR gene consists of many regulator complexes, proteins, and promoters that all assist in the expression of fluorescent proteins. First, there is the PR promoter, which regulates the XYLR exon itself. It is followed by the RBS, or ribosomal binding site, which orchestrates the timing and translation of the expression of the XYLR gene itself. Then comes the XYLR exon itself. The XYLR gene codes for an XYLR protein, triggers a secondary response in E. coli. It is important for there to be a double stop codon termination sequence that consists of a stem loop, and has both forward and reverse stoppage mechanisms in order for there to be no disruption of the reactions involved in xylene detection downstream of the XYLR exon. The secondary gene sequence that is affected by the XYLR protein consists of the PU promoter, which strictly regulates the YFP yellow fluorescent protein gene. And then there's another RBS, or ribosomal binding site, that orchestrates the timing of the YFP. In the case of xylene, if there is xylene, the bacteria will glow yellow, and the YFP will be expressed to indicate a positive test.
I'm Ollie. I was brought onto the iGEM team to help out with the engineering for synthetic biology. Synthetic biology has two aspects of its engineering. You have the traditional engineering and then the genetic engineering, which is the biological engineering. We were trying to, I came in and I helped design a device to implement our design. It's pretty much a breathalyzer in which the lung cancer patient breathes into it. The bag inflates with the lung cancer patient's breath and the VOC markers that would trigger our detection system. At the bottom of this bag is a well plate containing the bacteria that we have genetically engineered. Now, the bacteria that we have genetically engineered took a while to find the right genes that would react to the critical VOC markers that is in the lung cancer breath. And so, using some research and some other things, we came up with a few genes, and we have spliced them into some bacteria, E. coli, and with genetic engineering, and have provided us with the desired results. Next, the last part of this is receiving the correct signal from the bacteria, called transduction. So where we take the biological signal that the bacteria have given us, and we translate it into an electrical signal that we can read, which says that the patient has lung cancer. We were planning on using iGEM's 2009 Cambridge's team's idea of luminescence, adding an extra gene at the end of these bacteria in these E. coli, in order to give us a positive read, at which point we would know whether the patient had lung cancer or not.
Like a professional research lab, we had the idea of creating for the iGEM team applied research and fundamental research. Fundamental research tends to be the exploration of the basic ideas of biology and biotech. Applied research is using what we learn in order to use it for the goal, such as Howard's goal or any other endeavors that we might undertake.
The Applied Research Committee of the St. Mark's iGEM team this year has been really multifaceted. At the beginning, we started with 3A assembly, and we needed to make sure that we could master this with great efficiency and accuracy. Nearing the end of the 3A assembly, then we moved on to assisting the Fundamental Research Committee with their work, and we started researching genes whose translated proteins would potentially react with the VOCs found in lung cancer patients. By the end, when we identified our 3 main genes, we moved on to our next phase, which was the actual assembly and creation of our device. After finalizing the 3 genes that we wanted to use, which were FRMR, XYLR, and ALDB, we decided to start beginning the actual assembly of our new device. For this, we used the Gibson assembly and the 3A assembly, and this was the phase when we incorporated all the research that we did to put this in our actual device.
The St. Mark's iGEM team takes safety very seriously. We all use goggles, gloves, and lab coats when doing lab work. Our lab is fitted with safety equipment.
We decided to create a mission statement for the iGEM team so that all the members could know what the iGEM team stood for, and also so that we could have direction in the future.
To improve the community
by pioneering investigations
into the biotechnology.
By solving real-world problems in order
to promote scientific inquiry and educate
the future leaders of biological engineering.
The iGEM team, in accordance with its mission, has made it a point to not only do research in the lab, but also to reach out to the greater community.
We launched our school's greatest social media campaign through Facebook, Twitter, Instagram, and YouTube. We strove to inspire students to become passionate scientists and good leaders in tomorrow's world. Through social media, we were able to incite a global audience to action by showing them the potential synthetic biology has to solve some of the world's most pressing problems.
Creating a compelling web presence was one of our primary goals in both community and global outreach. We recognize that our website is an important medium to showcase our work to the world. It's a compilation of all our work in progress throughout the year that is available to a global audience. Through our wiki and other methods of outreach, we hope we can promote future scientific interest within our school and community.
The Applied Research Committee collaborated with the Outreach Committee to create instructional tutorial videos for the protocols and individual procedures of the 3A assembly. With our videos, we wanted to empower a global online audience to go and pursue their interest in biotechnology.
I think that the comic is important because it caters to an age group that's still very malleable, but also doesn't know very much about biotechnology. So I think the goal was to educate kids about biotechnology, our project, and the direct correlation between smoking and lung cancer, while also keeping it interesting for them to read. And so our main character was super biotech guy. So I hope it's interesting for them and will lead to a bright future for biotechnology.
The Brendan Court Program is a 4-week-long intensive program for DISD public school students who have been either in low-income situations or have not received the opportunities that other students in the Dallas area have received. These students are high-performing in their classes, but most of them have demonstrated financial need, and so the school provides a 4-week-long program for them to come over the summer free of tuition to learn and to continue to engage their minds. This iGEM team decided to take the initiative to prepare these students, my 8th grade students, for the real world of biotechnology. They showed them the dangers of smoking, and they also showed them the different kinds of research that they were doing. A lot of these kids had never seen a lab before. They'd never seen pipettes or stir rods or any of that. And thanks to our biotechnology team, we were able to expose them to the real world of biotechnology. For example, the idea of inserting DNA into a plasmid. That idea never would have made sense to a 12 or 13-year-old if we just tried to explain it. However, this iGEM team used PowerPoint presentations that were neatly animated in addition to this comic and other resources to ensure that these students would fully understand that in each bacterium there is a circle of DNA called a plasmid, that we cut out a section and that we insert our own gene. That idea is pretty complex for 7th and 8th graders to understand. Thanks to this iGEM team, they got it. It's very important to note that this program is tuition-free and runs entirely as a charity. This iGEM team was able to perform community service. They had such a strong desire to serve that they went out of their way. They spent their time, their resources, their money, and went out to provide this presentation for my students.
My name is Tim O'Meara. I'm one of the care captains of the iGEM team next year for St. Mark's. and I'm really looking forward to our opportunities in research. We've already developed the theoretical constructs for our genes and their uses, and I'm really looking forward to being able to put those into play and observing them and really working toward building the functional detector for lung cancer.
As future co-captain of the 2014-2015 St. Mark's iGEM team, I seek to build upon our current project this year and continue to screen VOC biomarkers in the detection of certain diseases like lung cancer. My ultimate goal is to promote scientific inquiry not only in the school community, but globally.
For millennia, we lay vulnerable to the capricious whims, the genetic mutations of the world.
We saw the forces of nature as insurmountable and unalterable.
But when we peered through early microscopes and found Mother Nature's cells, we saw that we could unlock them to steal, modify, and replace their sacred codes.
Now we peer at X-ray crystallographic images and through microscope lenses into the hidden worlds of our bodies.
As one of our awed Brendan Court science students said upon looking through a microscope for the first time,
there was an entire universe in there.
This universe is our playground where we thrive, where we mold and twist the swings and monkey bars to meet our needs and take us to where we want to go.
No longer do we slip along the rigid slides that Mother Nature proposes we take.
Instead, we take control and bend the plastic, which seemed set in stone a generation ago.
Just as we twist these slides,
we young researchers, in this age of budding biotechnology, yearn to find and warp our intellectual limit.
As a newcomer to the iGEM competition, we learned about the intricacies of synthetic biology, discovered new avenues for research, and shared our infectious optimism and indefatigable determination with the world. We kindled a fire for a better tomorrow and inspired hundreds to do the same. Our journey has only just begun, and we look forward to the groundbreaking strides our iGEM team will make in the future.